Welcome back to PharmaZone, A to Z of pharmacy, one stop where you can learn anything related to pharmacy and PharmD. In this particular video, we are going to begin with a new subject from second year of PharmD, that is pharmacology. The first chapter that we are going to explore is the introduction to autonomic pharmacology. As we all know, our nervous system is divided into the central and peripheral nervous system. The peripheral nervous system is further divided into the somatic nervous system and the autonomic nervous system. The autonomic nervous system is further divided into sympathetic and parasympathetic nervous system. Here we are focusing upon ANS and drugs acting upon ANS. The part of our nervous system that regulates the functions of visceral organs in the body is known as autonomic nervous system or ANS. Autonomic nervous system is not under voluntary control, that is, it is involuntary in its action, and therefore it was named so by Langley. Autos meaning self, nomos meaning governing in Greek. ANS innervates the heart, the smooth muscle, the glands, and the viscera and controls the functioning of these organs. As ANS regulates the functioning of visceral organs in the body, it plays a vital role in maintaining homeostasis of internal environment. It controls involuntary activities like digestion, heart rate, salivation, excretion, sexual arousal, etc. One more unique thing to note here is that ANS occupies a unique place because of its immense speed and intensity with which it regulates and balances the visceral functions. As we all know, ANS innervates the heart, the smooth muscle, the glands, etc. The centers for autonomic reflexes are present in the hypothalamus, medulla, and spinal cord. ANS consists of two major divisions like we have seen earlier, that is the parasympathetic system and the sympathetic nervous system. Most of the viscera have both sympathetic and parasympathetic innervations. The two divisions have opposing effects and normally their effects are in a state of equilibrium. The prime function of the sympathetic system is to help a person to adjust to stress and prepare the body for fight or flight reactions, while that of parasympathetic is to mainly participate in tissue building reactions. For example, like we can see in this image, the pupil of our eye gets constricted under parasympathetic system while it gets dilated under the sympathetic system. Although they have opposing effects, we can observe that they're complementary to each other. We can survive without the sympathetic system, that is, if we live a stress free life, but not without the parasympathetic system. In simple terms, we can understand that the sympathetic system is excitatory in its nature, while parasympathetic system is inhibitory in its nature. Now let's see the autonomic innervations. Autonomic nerves travel throughout our body to all the organs. The nerves are divided into the afferent and efferent nerves. The autonomic afferents, these are carried in visceral nerves through non-myelinated fibers. For example, the parasympathetic afferents are carried by the ninth and the 10th cranial nerves. These nerves relay sensory information from sensory receptors to region of brain. In simple, they conduct sensory impulses from internal organs and deliver it to the CNS. The second one is the autonomic efferents. Autonomic efferent innervation consists of a myelinated preganglionic fiber with synapses with the axon of a myelinated postganglionic fiber. These nerve fiber that exist at a region to act on muscle and glands or organs. The autonomic efferent innervation consists of a myelinated preganglionic fiber that synapses with the axon of a non-myelinated postganglionic fiber. The postganglionic fiber in turn forms a junction with the receptors of organs supplied by it. The ANS is unique in that it requires a sequential two neuron efferent pathway. The preganglionic neuron must first synapse onto a postganglionic neuron before innervating the target organ. The junction between the pre and postganglionic fiber is called a ganglion, and between the postganglionic fiber and the receptor is known as the neuroeffector junction. The traveling of an impulse along the nerve fiber is known as conduction, while its passage across a synapse is known as transmission. The autonomic efferent is divided into sympathetic and parasympathetic divisions. The parasympathetic efferent are carried through the craniosacral outflow and are located close to the innervated structures and hence are short. Outflow meaning that neurons begin at the cranial nerves. The sympathetic efferent extend from first thoracic to second or third lumbar segments of the spinal cord. They are found in the paravertebral, prevertebral, and terminal sites. These fibers are long and innervate the head, neck, viscera of thorax, and abdomen. Let's understand this pathway in detail. Parasympathetic or vagal nerves leave the vagal nuclei within the medulla as preganglionic efferent vagal fibers. These preganglionic efferent fibers are relatively long and do not synapse until they reach their target organ. The preganglionic fibers synapse within the small ganglia located near the target tissue and release the neurotransmitter acetylcholine, which binds to nicotinic receptors, causing depolarization and action potential generation in the short postganglionic vagal fiber that synapse at the target cells. Synaptic nerves that originate within the medulla travel down the spinal cord where they synapse with preganglionic cell bodies within the spinal cord. These cell bodies give off preganglionic fibers that are relatively short. These preganglionic fibers enter the paravertebral ganglia that are found on either side of the spinal column. These preganglionic neurons synapse with the cell bodies of postganglionic synaptic fibers. Acetylcholine is the neurotransmitter within these ganglia, and acetylcholine binds to nicotinic receptors on postganglionic neurons. From these neurons arise relatively long postganglionic fibers that travel to their target organ and release norepinephrine as the primary neurotransmitter, which binds to alpha or beta adrenoreceptors on the target tissues. Some of the preganglionic sympathetic fibers, instead of synapsing within the paravertebral ganglia, synapse in prevertebral ganglia located within the abdomen. Acetylcholine, which binds to nicotinic receptor, is a neurotransmitter at these sites as found in the other autonomic ganglia. Postganglionic sympathetic fibers then travel from the paravertebral ganglia to innervate tissues such as blood vessels, where they release norepinephrine as their primary neurotransmitter, which mainly binds to vascular alpha adrenoreceptors. Therefore, sympathetic postganglionic fibers can originate in either paravertebral or prevertebral ganglia. Now that we have learned in detail about the autonomic nervous system, let's understand that for the transmission of an impulse across a synapse, a neurohumoral transmitter substance is released into the sympathetic cleft. That is nothing but the neurotransmitters. The main neurotransmitters are acetylcholine, noradrenaline, and dopamine. The adrenal medulla releases the adrenaline and the noradrenaline. As far as the pharmacology of the autonomic nervous system and the drugs acting upon ANS is concerned, these are the classification of drugs acting upon ANS. We will be exploring this further in the upcoming videos. Until then, you can make a note of this, and meanwhile, like, share, and subscribe to PharmaZone. The notes are as usual available in the